Welcome back friends. Welcome back to another video session from this channel. So in this video session we will be studying regarding the mechanism, the process which is involved in organizing the urine formation in detail. So here the entire mechanism, the entire process which is involved in organizing the urine formation, it can be explained or it can be studied under three different strategies. And here these three different strategies include glomerular filtration, tubular reabsorption and tubular secretion. So what are those? Glomerular filtration, tubular reabsorption, and the other one is tubular secretion. So here, prior to the explanation of glomerular filtration in detail, at first if we consider the types of nephrons, basically nephrons are of two different types, and here these two different types of nephrons include cortical nephrons and the other one is juxtamedullary nephrons. So what are those? Cortical nephrons and the other one is juxtamedullary nephrons. So at first, if we consider the cortical nephrons, as the name itself suggests, so here in case of the cortical nephrons, the malphizian body, proximal convoluted tubule and as well as the distal convoluted tubule, they remain at the cortex region. But whereas the loop of Henle, it won't extend towards the medullary region. That means the size of the loop of Henle, it remains very shorter in case of the cortical nephrons. So here around 70 to 80% of the nephrons are cortical nephrons. 70 to 80% of the nephrons are cortical nephrons. And whereas the quite opposite scenario, it relates in case of the juxtamedullary nephrons. So here in case of the juxtamedullary nephrons, the malphizian body, the proximal convoluted tubule and as well as the distal convoluted tubule, they remain at the cortex region. But whereas the loop of Henle, it extends successfully towards the medullary region. So here the size of the loop of Henle, it remains so longer in case of the juxtamedullary nephrons. So the reason why these specific nephrons, they are termed as what? Juxtamedullary nephrons. So here around 15 to 20% of the nephrons are juxtamedullary nephrons. And as I have already mentioned you, the entire mechanism which is involved in organizing the urine formation, it can be explained under three different strategies. And here these three different strategies, they are nothing but what? Glomerular filtration, tubular reabsorption and tubular secretion. At first, if we consider the glomerular filtration, so here as the name itself suggests, so here the filtration mechanism or the filtration process, it occurs in case of the glomerulus. So the reason why here the specific strategy here it is termed as what? Glomerular filtration. So initially, if we consider the specific glomerulus, so here basically the specific glomerulus, it contains two different blood vessels. What are those? One is the afferent renal arteriole, which is having the wider diameter, and the other one is efferent renal arteriole, which is having narrow diameter. So here due to the variation in the diameter of the blood vessels, here it leads to the activation of a specific pressure, which is nothing but what? Ultrafiltration pressure. So what it is? Ultrafiltration pressure. So let me write it here. Ultrafiltration pressure. So initially the afferent renal arteriole, it brings the oxygenated blood from the renal artery. So here initially the renal artery, it is responsible for bringing the oxygenated blood from the right auricle of the human heart. So here once when the specific renal artery, it brings out the oxygenated blood from the right auricle of the human heart, now here the specific renal artery, it undergoes successful differentiation. So once when the specific renal artery, when it undergoes successful differentiation, it leads to the opening of afferent renal arteriole. As we all know, one human kidney, it contains some millions of nephrons. So here initially the specific renal artery, it brings out the oxygenated blood from the right auricle of the human heart. Then after the specific renal artery, it undergoes successful differentiation in order to form the efferent renal arteriole. Then after the specific efferent renal arteriole, it is responsible for inducing the oxygenated blood into the lumen of the glomerulus. Renal artery, it brings out the oxygenated blood from the auricle of the human heart. Now here the specific renal artery, it undergoes differentiation in order to form efferent renal arteriole. Efferent renal arteriole. So here once when the efferent renal arteriole, when it induces the oxygenated blood into the lumen of the glomerulus, so here within the lumen of the glomerulus, here there is an availability of bunch of blood capillaries, and here these bunch of blood capillaries, they are termed as what? Glomerular blood capillaries. So here these specific blood capillaries, they are responsible for interconnecting the two blood vessels which are nothing but efferent renal arteriole and as well as efferent renal arteriole. So here beneath the specific blood capillaries, here there is an availability of a specific membrane which is nothing but what? Basement membrane. So here beneath the basement membrane, here there is an availability of a specific layer which is nothing but what? Visceral layer. What it is? Visceral layer. Beneath the basement membrane, here there is an availability of a specific layer which is nothing but visceral epithelial layer. And here the visceral epithelial layer, it contains specialized cells which are nothing but what? Podocytes. So let me write it on the board. What it is? Podocytes. The visceral epithelial layer which is present beneath the basement membrane. So here it is nothing but the basement membrane. Beneath the basement membrane, here there is an availability of visceral epithelial layer, and here the specific epithelial layer, it contains specialized cells which are nothing but what? Podocytes. Podocytes. In between the two successive podocytes, here there is an availability of small specialized filtration slits. So here these specialized filtration slits or filtration pores, they are responsible for the passage of the various chemical components which are a part of the oxygenated blood. Is it clear? Initially, once when the oxygenated blood, when it successfully enters through the efferent renal arteriole, so here the specific oxygenated blood which successfully enters through the efferent renal arteriole, the oxygenated blood, it contains various chemical components. So here what are the various chemical components which are a part of the oxygenated blood here? The various chemical components which are a part of the oxygenated blood include glucose, amino acids, vitamins, electrolytes, hormones and as well as water. And in addition, it also contains certain blood cells and even it also contains the plasma proteins. So let me write it on the board. The chemical components of the oxygenated blood include chemical components of the oxygenated blood include glucose, amino acids, vitamins, electrolytes or ions, and in addition, even it also contains the water molecule, and in addition it also contains blood cells, and even it also contains the plasma proteins. So here all these are the various chemical components which are a part of the oxygenated blood here. So here once when the oxygenated blood, when it successfully enters through the efferent renal arteriole, so here beneath the blood capillaries, here there is an availability of basement membrane, and beneath the basement membrane, here the specific epithelial layer, here it is nothing but what? Visceral epithelial layer. So here the visceral epithelial layer, it contains many number of specialized cells which are nothing but what? Podocytes. In between the two successive podocytes, here there is an availability of small specialized pore-like structures which are nothing but what? Filtration slits or filtration pores. So here the specific filtration slits or filtration pores, they are very size specific. That means they won't allow all the types of molecules which are a part of the oxygenated blood. They are capable to allow only the macromolecules like glucose, amino acids, vitamins, electrolytes or ions, and as well as the water molecule. They won't allow the macromolecules like blood cells and as well as the plasma proteins. Is it clear? Because the specific filtration slits or filtration pores, they are very minute structures. They won't allow the macromolecules which are a part of the oxygenated blood like blood cells and as well as the plasma proteins. And the rest of the other chemical components which are a part of the oxygenated blood, they are capable to undergo permeabilization through the filtration slits or the filtration pores. So once when all these chemical components, once when they are capable to undergo permeabilization through the filtration slits or the filtration pores, now here it leads to the formation of a specific filtrate which is nothing but what? Glomerular filtrate. So here the glomerular filtrate, it contains all the various chemical components of the oxygenated blood except the blood cells and as well as the plasma proteins, and the rest of the other chemical components which are a part of the oxygenated blood like glucose, amino acids, vitamins and electrolytes and even the water molecule, they remain in the glomerular filtrate. So here once when the specific glomerular filtrate, when it is successfully formed, so here it is nothing but what? The glomerular filtrate, right? So here it is the entire glomerular filtrate. So here the glomerular filtrate, it remains in case of the capsular space. So here it is nothing but what? The Bowman's capsule. So the wider cup-shaped structure which is nothing but the Bowman's capsule. So within the Bowman's capsule, here there is an availability of a specialized space-like structure which is nothing but what? Capsular space. Within the capsular space, the entire capsular space, it is engulfed with the glomerular filtrate. So once when the glomerular filtrate, when it remains in case of the capsular space, now the specific glomerular filtrate, it is capable to transport from the Malpighian body into the renal tubule. So as we all know, the renal tubule, basically it contains three differentiated components. What are those? Proximal convoluted tubule, loop of Henle and as well as distal convoluted tubule. So here proximal convoluted tubule, or in other terms we can consider it as first convoluted tubule. So here the proximal convoluted tubule, it acts as an hotspot for undergoing the reabsorption of various chemical components like glucose, amino acids, vitamins, electrolytes and even the water molecule. Is it clear? All the various chemical components which are a part of the glomerular filtrate, they undergo reabsorption in case of the proximal convoluted tubule. So here you may get a small clarification why the specific glomerular filtrate initially which forms in case of the specific capsular space, why the specific glomerular filtrate, it undergoes reabsorption in case of the proximal convoluted tubule, because the glomerular filtration, it is an biased filtration. Is it clear? So let me write it on the board. Glomerular filtration, it is an biased filtration. So what is the meaning of biased filtration? That means the specific glomerular filtration, it won't distinguish the useful substances from the non-useful substances. So here both the useful substances and as well as the non-useful substances, they undergo filtration in case of the specific Bowman's capsule. So the reason why here the specific glomerular filtration, here it is termed as what? Biased filtration. So here the specific glomerular filtrate which remains in case of the capsular space, it contains both the useful substances and as well as the non-useful substances. So here these useful substances which are a part of the glomerular filtrate, they undergo reabsorption in case of the proximal convoluted tubule. So here what are the various chemical components which undergo reabsorption in case of the proximal convoluted tubule? Here the various chemical components include glucose, amino acids, vitamins and certain electrolytes like 70% of sodium, 70% of potassium and even it also contains the water molecule. Is it clear? So let me write it on the board. So let me arrange the things first. The various chemical components which undergo reabsorption in case of the proximal convoluted tubule include glucose, amino acids, vitamins and certain electrolytes like 70% of sodium, chemical components which undergo reabsorption in proximal convoluted tubule. So here these chemical components, they undergo reabsorption from the proximal convoluted tubule because all these chemical components, they are responsible for the sustenance of the living organism. So the reason why these specific chemical components, they undergo reabsorption in case of the proximal convoluted tubule. And here the various chemical components include glucose, amino acids, vitamins, and certain electrolytes like 70% of sodium, 70% of potassium, and even it also contains water molecule. So here the major portion of the water molecule, it undergoes reabsorption in case of the proximal convoluted tubule. And here the specific water reabsorption which occurs in case of the proximal convoluted tubule, here it is termed as what? Obligatory water reabsorption. Is it clear? So here the obligatory water reabsorption, it is a mandatory water reabsorption which occurs in case of the proximal convoluted tubule. Obligatory water reabsorption. Obligatory water reabsorption. So here you may get a small clarification how the specific water molecule, it is capable to undergo reabsorption in case of the proximal convoluted tubule because the epithelial layer of the proximal convoluted tubule, it contains microvilli. So here due to the presence of microvilli, so here in case of the proximal convoluted tubule, the water reabsorption, it is observed so accurately. Is it clear? And here all these are the various chemical components which undergo reabsorption in case of the proximal convoluted tubule. Then what about the various chemical components which undergo secretion in case of the proximal convoluted tubule? Here the various chemical components which undergo secretion in case of the proximal convoluted tubule include ammonia. Is it clear? Ammonia, creatinine, and even certain drugs. So let me write it on the board. The various chemical components which undergo secretion in order to form the nephric filtrate or the renal filtrate. Chemical components which undergo secretion. Undergo secretion in case of the proximal convoluted tubule. So these include ammonia, creatinine, and even drugs. So here why all these chemical components, they undergo secretion here? Because all these chemical components, they are very toxic to the human body. So the reason why these specific chemical components, they undergo secretion in order to form the nephric filtrate or the renal filtrate. Is it clear? So here once when all these specific chemical components, once when they undergo secretion, now here these specific chemical components, they enter into a specific component of the renal tubule which is nothing but what? Loop of Henle. Is it clear? So here the specific loop of Henle, here it is nothing but what? It is the U-shaped structure. So here basically the loop of Henle, it contains two specific components. What are those? So here it is nothing but what? The thin descending limb of the loop of Henle, and here it is nothing but what? The thick ascending limb of the loop of Henle. So here after the successful passage of the renal filtrate from the proximal convoluted tubule into the thin descending limb of the loop of Henle, so here the thin descending limb of the loop of Henle, it acts as an hotspot for the successful reabsorption of around 15 to 20% of water. Is it clear? 15 to 20% of water. The thin descending limb of the loop of Henle, it won't act as a hotspot for the reabsorption of any electrolytes. So here the thin descending limb of the loop of Henle, it acts as an hotspot for the reabsorption of only 15 to 20% of water. So here once when the specific water molecule, when it undergoes reabsorption, now here the specific renal filtrate or the nephric filtrate, it becomes much more concentrated. So here you make it a small clarification. When the specific water molecule, when it undergoes reabsorption in case of the proximal convoluted tubule and as well as the thin descending limb of the loop of Henle, how the specific nephric filtrate, it becomes much more concentrated. So here the answer to this question is very simple. So here in order to understand this specific concept, let me give you one common example what generally we experience in our daily life. So here let us consider one specific solution which is nothing but the sugar solution. So here after preparing the sugar solution, if we place the sugar solution on the surface of a Bunsen burner and when we heat the specific sugar solution, then after the level of the sugar solution gradually decreases because the water which is present in case of the sugar solution, when it undergoes evaporation, then after the levels of the sugar solution, it goes on decreasing gradually. So here once when the water levels in case of the sugar solution, when it goes on decreasing gradually, now here the sugar solution, it becomes much more concentrated in comparison to the initial times. Is it clear? So here the specific sugar solution, it becomes much more concentrated because of what? Because of the reduction in case of the water levels. So here if we apply the same concept here, easily we can understand. Initially the specific glomerular filtrate, it contains much more water molecules. But once when the specific water molecule, when it undergoes reabsorption in case of the proximal convoluted tubule and as well as the thin descending limb of the loop of Henle, now here the specific nephric filtrate, it becomes much more concentrated. Is it clear? So here once when the specific nephric filtrate, when it becomes much more concentrated, now here the specific nephric filtrate from the thin descending limb of the loop of Henle, it successfully transports. Now here it reaches to the thin ascending limb of the loop of Henle. Is it clear? So here after reaching to the thin ascending limb of the loop of Henle, now here the specific nephric filtrate, it successfully reaches to the thick ascending limb of the loop of Henle. So here the thick ascending limb of the loop of Henle, it acts as a hotspot for the reabsorption of certain ions or electrolytes like sodium, chloride, magnesium and as well as potassium. Is it clear? What are the various ions here which undergo reabsorption? Sodium, potassium, chloride, magnesium. So here all these are the various ions or electrolytes which undergo reabsorption in case of the thick ascending limb of the loop of Henle. So here the specific sodium ion, it is capable to undergo active reabsorption. That means by taking the advantage of energy, here the specific sodium ion, it is capable to undergo reabsorption in case of the thick ascending limb of the loop of Henle. Because as we all know, the concentration of sodium, it is higher outer side and it is lower inner side. So here once when the specific sodium ion, when it travels against its concentration gradient from lower to higher, under such condition, the specific sodium ion, it takes the advantage of energy. So here by taking the advantage of energy, here the specific sodium ion, it is capable to come against its concentration gradient. So the reason why here the specific absorption of sodium, it is nothing but what? Active reabsorption. Is it clear? So let me write it on the board here. Sodium reabsorption, it is an active reabsorption which is observed in case of thick ascending limb of the loop of Henle. And in addition to the sodium and as well as potassium, even the chloride and as well as magnesium, they undergo reabsorption in case of the thick ascending limb of the loop of Henle. So here in case of the thick ascending limb of the loop of Henle, water molecules, they won't undergo reabsorption. Is it clear? Water reabsorption, it occurs only in case of the proximal convoluted tubule, and in addition the water reabsorption even it is observed in case of the thin descending limb of the loop of Henle. Water reabsorption, it won't occur in case of the thick ascending limb of the loop of Henle. Thick ascending limb of the loop of Henle, it acts as an hotspot for the reabsorption of only certain ions or electrolytes like sodium, potassium, chloride and as well as magnesium. So here once when all these ions or electrolytes, once when they undergo reabsorption in case of the thick ascending limb of the loop of Henle, now here the specific nephric filtrate, it becomes much more concentrated here. Is it clear? Then after the specific nephric filtrate from the thick ascending limb of the loop of Henle, it successfully transports to the distal convoluted tubule. So here it is nothing but what? The distal convoluted tubule, or in other terms we can consider it as second convoluted tubule. So here the distal convoluted tubule, it acts as a hotspot for the reabsorption of hydrogen carbonate. Is it clear? In general, from the distal convoluted tubule, hydrogen carbonate and as well as water molecule, they undergo reabsorption. So here distal convoluted tubule, most commonly it acts as a hotspot for the reabsorption of only hydrogen carbonate. So let me write it on the board. Let me arrange the things first. Distal convoluted tubule, it acts as a hotspot for the reabsorption of only hydrogen carbonate. Water reabsorption, it is not observed always in case of the distal convoluted tubule. So let me write it on the board. Distal convoluted tubule or second convoluted tubule. So here in case of the distal convoluted tubule, hydrogen carbonate and in addition water molecule, they undergo reabsorption here. Reabsorption. Is it clear? So here in general, water reabsorption, it is observed only in case of proximal convoluted tubule and as well as the thin descending limb of the loop of Henle. Distal convoluted tubule, it won't act as a hotspot for the reabsorption of water always. But under certain circumstances, water reabsorption, even it is observed in case of the distal convoluted tubule. And here the specific water reabsorption which is observed in case of the distal convoluted tubule, here it is termed as what? Facultative water reabsorption. So let me write it on the board what it is. Facultative water reabsorption. So here the facultative water reabsorption, it is not observed always, only under certain circumstances. So here the water reabsorption, it is observed in case of the distal convoluted tubule. So the reason why the specific water reabsorption, here it is termed as what? Facultative water reabsorption. So here the facultative water reabsorption, it is organized by the means of a specific hormone which is nothing but what? Vasopressin or antidiuretic hormone. That means under humid conditions, most of the water, it undergoes secretion in the form of sweat. And here some amount of water, it undergoes secretion in the form of urine. So here when the water levels in the body, when it decreases drastically, so here in order to overcome the dehydration mechanism or the dehydration process, initially when the levels of the water decreases gradually, under such condition, the volume of the blood in the body, it also decreases. So here once when the volume of the body in the blood decreases, instantly the osmoreceptors which are present in the hypothalamus of the forebrain, they undergo activation. So once when the osmoreceptors in the hypothalamus of the forebrain, once when they undergo activation, now here the specific osmoreceptors, they in turn they trigger or they activate the posterior pituitary. Is it clear? So let me write it on the board so that easily you can understand. Initially, when the water levels in the body decreases gradually, instantly the volume of the blood in the body decreases. Volume of the blood in the body decreases gradually. So let me write it on the board. Volume of the blood decreases. So once when the volume of the blood decreases, now here it leads to the activation of osmoreceptors which are present in the hypothalamus of the forebrain. So here the osmoreceptors in turn they trigger or they activate the posterior pituitary. Posterior pituitary. Posterior pituitary. Now here it is responsible for the secretion of a specific hormone, which is nothing but what? Vasopressin, or in other terms we can consider it as antidiuretic hormone. Is it clear? So here the specific vasopressin or antidiuretic hormone, it is responsible for activating the aquaporins or the water channels which are present in case of the distal convoluted tubule. So here these are nothing but what? Aquaporins are the water channels. So here in the presence of vasopressin or antidiuretic hormone, once when the aquaporins are the water channels, once when they undergo activation, from these aquaporins are the water channels, so here the water reabsorption takes place. So here this specific reabsorption of water, here it is termed as what? Facultative water reabsorption. So here in most of the cases water reabsorption it is observed only in case of the proximal convoluted tubule and as well as the thin descending limb of the loop of Henle. And here the water reabsorption it is not observed in case of the distal convoluted tubule always, but only under certain circumstances even the water reabsorption even it is observed in case of the distal convoluted tubule, and here it is termed as what? Facultative water reabsorption. Is it clear? So here once when the water reabsorption and as well as hydrogen carbonate when they undergo reabsorption in case of the distal convoluted tubule, now here we should have a basic idea regarding the peritubular network. As I have already mentioned you, the efferent renal arteriole which is having the narrow diameter. So here the specific efferent renal arteriole, it leads to the further opening of a specific peritubular network. So here the peritubular network which successfully originates from the efferent renal arteriole, it is widely spread out all the various components of the renal tubule. Is it clear? So let me write it on the board. So here it is nothing but what? The peritubular network which proceeds in the opposite direction. Peritubular network or the vasa recta. Is it clear? So here once when the specific peritubular network or the vasa recta, when it is capable to proceed very adjacently to the loop of Henle, now here it leads to the activation of a specific mechanism which is nothing but what? Counter current mechanism. Is it clear? What it is? Counter current mechanism. So here the specific mechanism through which the concentrated urine it successfully forms in case of humans, here it is nothing but what? Counter current mechanism. So let me write it on the board what it is. Counter current mechanism. Counter current mechanism. So here what is the meaning of counter current mechanism? So here in case of the counter current mechanism, exchanging of ions or electrolytes takes place in opposite direction. So here the specific mechanism, here it is nothing but what? Counter current mechanism. So let me write it on the board. So here in case of the counter current mechanism, exchanging of ions or electrolytes takes place in opposite direction. Exchange of ions or electrolytes takes place in opposite direction. So here presence of the counter current mechanism, here it leads to the formation of concentrated urine. Humans are capable to form nearly 4 times of concentrated urine in comparison to the initial glomerular filtrate because of the presence of counter current mechanism. Is it clear? So here what exactly happens in case of the counter current mechanism is exchanging of ions or electrolytes takes place in opposite direction. So here if we consider, as I have already mentioned you, the efferent renal arteriole which is having the narrow diameter, it leads to the opening of a specific peritubular network. So here the specific peritubular network, it is widely spread out in all over the components of the renal tubule. Is it clear? So if we look at here it is nothing but what? The thick ascending limb of the loop of Henle. Very adjacent to the thick ascending limb of the loop of Henle, here there is an availability of thin descending limb of vasa recta. Is it clear? So here the peritubular network, when it successfully reaches to the loop of Henle, or to the medullary region, here it is termed as what? Vasa recta. So what it is? Vasa recta. So here the vasa recta, it successfully passes out very adjacently to the loop of Henle. So here it is nothing but what? The thick ascending limb of the loop of Henle. Very adjacent to the thick ascending limb of the loop of Henle. Here there is an availability of thin descending limb of vasa recta. So here in case of the ascending limb of the loop of Henle, so here the successful passage of the nephric filtrate, it takes place or it occurs in case of the upward direction. But whereas in case of the thin descending limb of the vasa recta, in case of the thin descending limb of the vasa recta, the successful passage of the blood, it takes place in case of the downward direction. In case of the downward direction, right? So in this way. So here once when the specific blood, when it proceeds or when it travels in opposite direction, here as I have already mentioned you, very nearer to the thick ascending limb of the loop of Henle, here the sodium ion, it undergoes active reabsorption. Now here the specific sodium ion which undergoes active reabsorption in case of the thick ascending limb of the loop of Henle. Now here the specific sodium ion, it undergoes secretion. Now here the specific sodium ion, it undergoes secretion into the thin descending limb of the vasa recta. Is it clear? So here once when the specific sodium ion, when it undergoes secretion in case of the thin descending limb of the vasa recta, now here the specific nephric filtrate, it proceeds in this direction. In this direction, right? So here after proceeding in this direction, now here it is nothing but what? Here it is initially the thin descending limb of the loop of Henle. Very adjacent to the thin descending limb of the loop of Henle, here there is an availability of thick ascending limb of the vasa recta. So here after reaching to this particular point, now here the specific sodium ion initially which undergoes secretion, here in case of the thin descending limb of the vasa recta, now here it undergoes reabsorption very nearer to the thin descending limb of the loop of Henle. So let me write it on the board. So here the sodium ion, it undergoes reabsorption very nearer to the thin descending limb of the loop of Henle from the specific vasa recta. So here once when the specific sodium ion, when it undergoes reabsorption very nearer to the thin descending limb of the loop of Henle, now here it leads to the increase in, or here it leads to the increase in the concentration of medullary interstitial fluid. Is it clear? So here once when the medullary interstitial fluid concentration increases enormously after the successful reabsorption of sodium ion from the specific vasa recta, now here exactly what happens is the specific urea, it undergoes successful secretion at the thin ascending limb of the loop of Henle. Is it clear? So here it is nothing but what? The thin ascending limb of the loop of Henle, and here it is nothing but what? The thick ascending limb of the loop of Henle. So here the specific urea, it undergoes secretion into the thin ascending limb of the loop of Henle. So here after undergoing secretion in case of the thin ascending limb of the loop of Henle, now here the specific urea, it proceeds in upward direction. Now here from the ascending limb of the loop of Henle, now here the specific urea, it successfully enters into the distal convoluted tubule. After reaching to the distal convoluted tubule, now here the specific urea and as well as the water molecule, it undergoes successful secretion into the collecting duct. Is it clear? So here once when the specific water molecule and as well as the urea, so once when they enter into the collecting duct, now here the specific water molecule and as well as the urea, now here they undergo successful reabsorption from the collecting duct. Water and as well as urea, they undergo reabsorption. Is it clear? Initially the specific urea, it undergoes secretion into the thin ascending limb of the loop of Henle. So here after undergoing secretion, now here the specific nephric filtrate, it proceeds in upward direction. And here it successfully reaches to the distal convoluted tubule. From the distal convoluted tubule, now here it successfully reaches to the collecting duct. After reaching to the collecting duct, now here the specific water molecule and as well as the urea, it undergoes reabsorption. So here once when the counter exchange or the opposite exchange of ions or electrolytes takes place, it leads to the increase in the concentration gradient, or here it leads to the increase in the medullary interstitial fluid. Is it clear? Initially, the concentration of the medullary interstitial fluid, it relies around 300 osmolarity inverse. Is it clear? But gradually, the concentration of medullary interstitial fluid, it reaches to 600 osmolarity inverse. Then after, it reaches to 900. Then after, it reaches to 1200 osmolarity inverse. So here it is because of what? Because of the counter exchange of ions or electrolytes. So here, once when the concentration of the medullary interstitial fluid, when it increases enormously because of the counter exchange of ions or electrolytes in opposite direction. Now here, the specific nephric filtrate which has initially formed, now here it is capable to pass through the collecting duct. From the collecting duct, now here it reaches to the renal calcisis. Renal calcisis. From the renal calcisis, it reaches to the pelvis. From the pelvis, it reaches to the ureters. From the ureters, it reaches to the urinary bladder. So here, once when the specific nephric filtrate, when it successfully reaches to the urinary bladder, now here it remains in the formation of urine. So here, from the urinary bladder, the specific urine, it successfully reaches outer side by the means of urethra. So here, in this way, the concentrated urine, it is successfully formed in case of the humans because of the presence of a specific mechanism which is nothing but what? Counter current mechanism. So here in case of the counter current mechanism, successful exchange of ions or electrolytes takes place in opposite direction. So here urinary excretion, in simpler terms, urinary excretion, here it is nothing but what? Glomerular filtrate minus tubular reabsorption plus tubular secretion. So here it is nothing but what? Urinary excretion. So let me write it on the board. Urinary excretion, here it is nothing but what? Urinary excretion. It is nothing but the glomerular filtrate. Glomerular filtrate minus tubular reabsorption plus tubular secretion. So here it is nothing but what? The urinary excretion. Is it clear? And here if we consider the net filtration pressure, net filtration pressure, so here the total volume of the blood in the human body, here it is around 5 to 6 liters. 5 to 6 liters of human blood. Is it clear? And for 1 minute, per 1 minute, here the specific human excretory system, it is capable to form around 125 milliliters of glomerular filtrate. Is it clear? For 1 minute, the human excretory system, it is capable to form around 125 milliliters of glomerular filtrate. And for 1 day or for 24 hours, the specific human excretory system, it is capable to form around 180 liters of glomerular filtrate. 180 liters of glomerular filtrate is formed. Is it clear? So here this is regarding the specific mechanism, the specific process which is involved in organizing the urine formation in case of humans. Is it clear? So here I hope that this video will help you a lot. So if you like this video, just hit the like button and share it to your friends.